Greetings respected judges and fellow peers. Today we are going to present our review entitled Employing Omics in Cancer Drug Discovery, Recent Updates towards Personalized Medicine for Lung Cancer. But before that, let us introduce ourselves first. My name is Ciputra and two of my colleagues are Nathaniel, Alvin, Sanjaya and Alvaro, Filbert, Liko. Lung cancer has been one of the most prevalent types of cancer worldwide. Especially in Indonesia, it has become the first and fifth leading cause of cancer-related death in Indonesian men and women respectively. Despite the advance of medical sciences, we still do not see any significant decrease in the number of lung cancer-related deaths in Indonesia. This raises a question: what is the issue with current management of lung cancer in Indonesia? Pathologically, the major concern of lung cancer is its genetic heterogeneity. As we can see in this pie chart, there are various types of lung cancer mutation occurring in Asian populations. Unfortunately, the current approach for lung cancer management relies on the common use of drugs for lung cancer patients with different genetic alterations. This is not appropriate since it can lead to off-target side effects and further complications that may lead to mortality. Omics analysis has significantly improved our understanding of the lung cancer molecular alterations over the last decade. It is a systemic molecular profiling of patient-by specimen which allow personalized and complete molecular characterizations by identifying genotype-phenotype relationships. The presence of omics analysis can be used to identify patient-specific drug targets for treatment optimization, thus allowing a more personalized approach of medicine. Even though this strategy is considered promising, the standard workflow of our omics-based analysis targeted therapy in lung cancer is still unclear. Therefore, in this literature, we aim to identify and review the molecular signature of lung cancer based on omics analysis and currently available targeted therapy to construct the standardized pipeline for omics implementations in lung cancer targeted therapy in Indonesia. To do that, we perform scientific literature review by using Google Scholar search engine. We use lung cancer, omics, and targeted therapy for the keywords to look for research paper, literature review, short comments, systematic review, and meta-analysis published from 2010 to 2020. The inclusion criteria for the paper selection were related to current targeted therapy for lung cancer and molecular signature of lung cancer based on multi-omics analysis. Then, data analysis was conducted to construct general pipeline for omics implementations in lung cancer targeted therapy and drug discovery in Indonesia.
For the result, we gathered several consistent molecular signatures from available omics studies. These include several aberrations in genomic, transcriptomic, proteomic, meronomic, metabolomics, and cytomics level. Different patients should have different molecular alteration at specific level. It is also possible for different patients who have the same molecular signature at one level while having different molecular signature at other levels. It creates combination of molecular signature which contribute to the complexity of lung cancer itself. Our lack of knowledge about this multi-level molecular alteration is causing the ineffective management of lung cancer. On the other side, these various molecular signatures also create opportunities to be exploited as promising drug targets. Unfortunately, we found that current targeted therapies are mostly dominated by tyrosine kinase inhibitor and immunotherapy-based drug. This is unfortunate because, based on the omics analysis, a broad spectrum of targets were revealed, which means it is yet to be explored. We also found that natural-based therapies, drug research used in lung cancer, were actually none from Indonesia, despite our country-rich biodiversity. To optimize the management of lung cancer, we can implement omics analysis for targeted therapy and drug discovery purposes. For validation of targeted therapy, we can follow the pipeline right here. The first step is to collect the biospecimen from patient, followed by multi-omics analysis that can be conducted at different levels to identify the conserved molecular signature from biological specimen. Well, for biological data, such as radiological image, instead of identifying the conserved molecular signature between patients, we identify the whole tumor microenvironment and heterogeneity by using deep learning system and image processing. The last step is to integrate the multi-omics analysis for drug validation purposes that can be used to prescribe treatment based on the already mapped molecular signature.
Figure 3 shows the pipeline of Omics analysis implementation in drug discovery in Indonesia. There are two aspects to be considered, which are natural anti-cancer product library preparation and molecular profiling of Indonesian population, which both are yet to be developed. High-throughput screening can be conducted from Indonesian biodiversity resources to identify promising anti-cancer natural products. These compound libraries can be integrated with Indonesian lung cancer patient and genetic databases to specifically utilize the natural product to address lung cancer in Indonesia. Henceforth, integration of omics study to the drug discovery process allows the optimization of ethnopharmacological research output in Indonesia. In summary, omics analysis has a huge potential as our primary vehicle in the development of ultimate targeted therapies for lung cancer. It allows shifting of drug target focus from just tyrosine kinases to the complete molecular profile. By integrating comprehensive natural compound databases with high-throughput patient data management, Indonesian biology-based ethnopharmacological research for cancer can be maximized. And that concludes our poster presentation. Thank you.